السلام عليكم. أنا الطالب ركان حداد من كلية الطب في الجامعة الأردنية. Today we're going to be studying the fifth lecture of Neoplasia. First of all, we must understand that this lecture is not going to be as hard as the previous lectures. Hopefully it's going to be an easy, quick lecture to study. Let's first of all talk about insensitivity to growth inhibitory signals. First of all, there are signals inside of our body. They're called the growth inhibitory signals. What do? طبعا شو عملت بس قصيت هيك الشاشة. Anyways, they are growth inhibitory signals. What do? They inhibit growth. They inhibit excessive growth. What is a gene that is basically responsible for that? The tumor suppressor genes. Okay, so tumor suppressor genes, they have growth inhibitory signals. Now, cancer cells, they have insensitivity to these signals. Okay, so really simple concept. Normally, there are growth inhibitory signals. Lakin, because of cancer, it becomes insensitive to these signals. Okay. Now, tumor suppressor genes, they have the anti-growth signals or the growth inhibitory signals. It prevents cellular growth in many mechanisms. منهم, it may cause the dividing cell to enter the quiescent stage. To the quiescent stage or the quiescent state هو G0. We know that the cell cycle هي عبارة عن G1 S, okay, G1 S, then the G2. بعد الجي 2 بيجي هو الميتوتيك phase اللي بصير فيها the splitting of the cell. Okay? Now, G1 to S, this is a really important transformation that is governed by lots of genes, by lots of whatever. Okay? Something that is checkpoints and so on. Something that is really important is the quiescent state, G0. The cell might enter the quiescent state. Hold on, the cell cannot do anything that has to do with cellular proliferation. The cell will not proliferate, okay? And this is what the tumor suppressor gene does to cells that might form excessive growth. It tells it, no, you have to stop here, no more growth. As well as it causes the re-entry of cells in a post-mitotic differentiated pool, and it will lose replicative potential. بكل بساطة, the tumor suppressor gene is going to notice that a cell is trying to proliferate in an excessive way. It holds it, it tells it, no, you cannot move any further. I want you to stop. You're not going to ever undergo mitosis. I'm just going to shut you down for good, okay? I will put you in a differentiated pool. You're a differentiated cell that must not proliferate. You stay here, okay? Now, another important gene is the retinoblastoma gene, the RB gene. We talked about it as well. We mentioned how it's a really common gene to get mutated in cancers. The majority of cancers often will find a retinoblastoma gene to be mutated. Why? Because it's the governor of the cell cycle. Okay? It governs the cell cycle. بتدير بالها على الcycle نفسها. It takes care of it. Okay? So it's a negative regulator of the cell cycle. If there are lots of cell cycles occurring, lots of mitotic activity, the RB gene, the retinoblastoma gene, will regulate it negatively. Yani it will inhibit the activity of the cell cycle. Okay? Now, when it comes to mutations will ruin the activity of the RB gene. طب شو ال RB gene بيعمل? The RB gene basically stops the cell cycle. So if it's mutated, the cell cycle will never stop. Alongside other factors that affect the cell cycle. Okay? Now, this mutation can either directly or indirectly inactivate these, directly or indirectly inactivated in the most human cancers. Okay? The RB gene is either directly or indirectly inactivated. Okay? When the RB gene is activated, everything is healthy, everything is working, the cell cycle is stopped when it's trying to move on excessively. Lakin when a mutation occurs or when a cancer occurs, the RB gene will directly or indirectly be inactivated. Okay? It's really important to understand that. طبعا the RB gene, I repeated that by accident. I was typing it really late at night, so I'm really sorry about that. The retinoblastoma RB gene, the first and prototypic cancer suppressor gene to be discovered. It was the first one to be discovered to have caused cancer, because mutations in it cause cancer. The first one to be discovered was retinoblastoma gene. It is an uncommon childhood tumor, okay? Approximately 60% of retinoblastomas are sporadic and 40% are familial. Okay, so 60% of them are sporadic and they occur due to natural مش natural يعني they occur due to other reasons يعني. And 40% اللي هم familial هذول إنه there is a genetic trait one. Okay, the predisposition to develop the tumor being transmitted as an autosomal dominant trait. Okay, شو حكينا هون؟ autosomal dominant trait. يعني the RB gene is not on it's not X-linked or Y-linked, it's autosomal يعني on a على chromosome جسمي يعني نحكي له. Okay, and basically the ability to develop the tumor, the mutation has to be dominant. كيف يعني dominant؟ يعني لازم يصير في mutation على 2 alleles. Okay, هلا we know that let's just يعني talk about something really quickly. An allele is basically a letter of the gene. Let's say a gene has 2 alleles, A and A. Okay, this means that the trait is going to appear as A. What if it's A capital and A small? Also A because it's dominant. Okay, so let's consider this as the RB gene. Okay, if one mutation occurs and it changes the capital A to a small a, nothing will happen. Okay, nothing will happen. But if 2 mutations occur, it will change to small a, small a. Here the RB gene is going to be mutated. Okay, here the RB gene is going to be mutated. فنحكي لك it's an autosomal dominant trait. Okay, to develop the tumor. To account for the sporadic and familial occurrence of an identical tumor, Knudsen in 1974 proposed his now famous 2-hit hypothesis. يعني شو يعني? 2 mutations are required to produce retinoblastoma. Retinoblastoma هو التومر نفسه. Okay? So, كم mutation لازمة؟ هم 2 mutations are needed. Okay? To produce that retinoblastoma. مش بس 1 mutation or 1 hit. لا لازم 2 mutations يعني 2 hits are needed or are required to produce the retinoblastoma. Okay. طبعا these involve obviously the RB gene that is located on chromosome 13q14. Okay, chromosome 13, position 14 of the gene. Both of the normal alleles of the RB locus must be inactivated. يعني 2 hits لازم يصيروا. Okay, as we said مش بس واحد من alleles, not just one allele has to be mutated. Both of the normal alleles must be inactivated or mutated. ليش؟ because the dominant trait, okay, the dominant trait is basically the زي ما هلا حكينا the A capital. Okay, the A capital is a dominant trait. This is the normal trait. Okay, هيك the person is normal. Okay, هاي هون there's no retinoblastoma. Okay, there's no retinoblastoma. Okay, you get it. There's no retinoblastoma. But what if one of them is mutated? برضو there's still no retinoblastoma. Only this was mutated. So what if both of them were mutated? So كانت AA and due to a mutation صاروا A small. This mutation هون تعمل retinoblastoma. Okay? So again, Knudsen, he proposed that 2 hits or 2 mutations are required to produce retinoblastoma. Okay? So both of the normal alleles, they have to be mutated. Not just one of them, both alleles must be mutated. Okay? In familial cases, children inherit one defective copy of the RB gene in the germline, while the other copy is normal. In familial cases, يعني we're talking about مثلا, let's say, a father that has the retinoblastoma and a mother who is healthy, so A small and A capital. They will eventually get a child that gets one allele from the father and the other from the mother. Okay, so basically this child here, they don't have retinoblastoma, but they inherited one mutated gene. Okay, يعني they inherited one mutated gene. This means that they only need one somatic mutation or one hit to produce retinoblastoma. Okay, فإن familial cases يعني what happens is you would need one mutation. Why would we need one mutation? Because already there is one allele that is defective. Okay, really simple concept. ما فيش إشي صعب. Okay, so retinoblastoma develops when the normal RB gene is lost in retinoblast as a result of the somatic mutation. زي ما حكينا. In familial cases, retinoblastoma families, only one mutation is required. ليش? Because the child already took a mutation or took a defective gene from a parent and a normal gene from another parent. Okay? So only one somatic mutation is needed to change the remaining allele. Okay? The familial transmission follows an autosomal dominant inheritance pattern. In sporadic cases, both normal RB alleles are lost by a somatic mutation, in one of the retinoblasts. In sporadic cases مش familial, يعني sporadic, مثلا a person who doesn't have retinoblastoma as a familial history, suddenly he gets retinoblastoma. ليش؟ لأنه two mutations have occurred. Okay, both the normal alleles have undergone a somatic mutation. Okay, really easy. A retinal cell that has lost both of the normal copies of the RB gene becomes cancerous. A retinal cell يعني خلية بالعين. Okay, that has lost both of the normal copies of the RB gene. شوف الميوتيشن وين صارت. It happened to a retinal cell. In that retinal cell, the mutation caused the loss of both normal alleles, okay, and both of the normal copies of the RB gene. It becomes cancerous. Although the loss of normal RB genes was discovered initially in retinoblastomas, it is now evident that homozygous loss of this gene is a fairly common event in several tumors. Okay, so basically what's happening here is that, as we said, a retinal cell can get mutated. In this mutation, this mutation can ruin the RB gene. If it basically causes the loss of both the normal copies, the retinal cell can become cancerous. Okay? Now, RB genes, the loss of the normal RB genes was only discovered in retinoblastomas. But they were able to identify a mutation in the RB gene in other cancers. Okay? So the RB gene is not only related to retinoblastoma, مع انه it's talking about the retina and the eye and whatever. لكن retinoblastoma gene mutation can occur to many other cancers. Okay? يعني we can find it with breast cancer, small cell cancer of the lung, and bladder cancer. Okay? So basically, patients with breast cancer, if we want to study the RB gene, it will be mutated. Okay? It will be lost. The normal copy will be lost. Small cell cancer of the lung, it will be lost. Bladder cancer, same. Okay? So patients with familial retinoblastoma also are at greatly increased risk of developing osteosarcomas and some soft tissue sarcomas. Okay? Why is that? Because they are more able to get, it's easier for their RB gene to be lost or mutated. Why? Because they already have a defective allele. Okay? So in familial cases or in familial retinoblastoma, they're already affected by one mutation. They only need one more. So they're more at risk of developing osteosarcomas and some soft tissue sarcomas. The function of the RB protein is to basically regulate the G1S checkpoint. شو هي G1S checkpoint? It's basically the checkpoint that tells the cell to move from G1 to the S in the cell cycle. Okay? So what regulates that? The RB protein. البروتين بيجي من الجين. So a normal RB gene, it basically gives a normal RB protein. تمام. And a normal RB protein is going to regulate the movement from G1 to the S in the cell cycle. Okay? So imagine that there's a mutation in the RB gene. This means, so if there's a mutation in the RB gene, this means that the RB protein is going to be mutated. ممكن ما ينتج أصلا. It either gives me a defective protein or no proteins will form أصلا. And this will basically خلينا نحكي stop governing the cell cycle. Okay, the cell will keep on moving from G1 to S without being regulated. Okay, and this will basically increase the growth one. Okay, the growth is gonna increase. Okay, so RB protein, it basically regulates how fast a cell can move from G1 to S. Okay. Now, if it's mutated, it won't be able to regulate, so the cell will do whatever it wants. تمام? It's really easy. Okay, the cell must pass, obviously, before the DNA replication commences, as a transition from G1 to S is an extremely important checkpoint in the cell cycle. Obviously, this picture is going to be repeated at last. Okay, هون we're looking at a sporadic form of the mutation. شو يعني sporadic؟ هاي somatic cells of the parents طبعا, and these are normal cells. يعني the RB gene is normal. Okay, the RB gene is normal. هاي جت الgerm cell وجت الovule مثلا, and it gave a zygote, and this zygote doesn't have a mutated RB gene. It's healthy. تمام. Anyways, هاي somatic cells of the child كلهم طلعوا normal. Okay. So هون بتشوف انه one mutation happened. Will that cause retinoblastoma? لا. تمام. It will not cause retinoblastoma. لكن when more mutations occur and both copies of the gene are mutated, then this means the person is going to have retinoblastoma. Okay, so it's really simple. هاي هي sporadic form, pathogenesis of retinoblastoma in the sporadic form. يعني the mutation occurred later on. It doesn't have to do with anything familial, it doesn't have to do with genetics or inheritance. لا ملهاش دخل. Okay, the sporadic form, the mutations occurred randomly throughout the person's life. Now, familial form, one of the parents has a mutant RB gene. The other doesn't have anything. Okay, so here we have a normal RB gene, here we have a mutated RB gene. When they produce a zygote راح يكون في a mutated copy and a non-mutated copy. تمام؟ كتير موضوع سهل. So basically, the somatic cells of the child are already going to have a defective gene. Okay, one of the genes أصلا is defective, the other is normal, لكن the other is defective. For only one mutation now is needed. شايفين هون it needed 2 steps of mutations. Okay, 2 hits, one to each gene. هون only one was needed to produce retinoblastoma. Okay, so بكل بساطة لازم نعرفه in the sporadic form 2 hits are needed, in the familial form 1 hit is needed. تمام? Okay. Again, let's talk about the relationship of the RB gene and the cell cycle. We just said how important it is for the G1S checkpoint, especially the RB protein. The RB gene product, اللي هو البروتين, is a DNA binding protein that is expressed in every cell type examined. It exists in an active hypophosphorylated and an inactive hyperphosphorylated states. إذا ننتبه على شو موجود عندنا. ال RB gene product هو عبارة عن DNA binding protein. Okay, so the gene that we have for the retinoblastoma اللي موجود أصلا على ال DNA, it will give you a specific protein. هاي ال RB protein. This protein is a DNA binding protein. It will go back and bind to a specific part of the DNA. تمام? And we can actually notice the RB protein in almost every cell type examined. Okay? هلا, when is it activated? When it's hypophosphorylated. يعني it doesn't have much phosphate groups to it. يعني إذا كان عليها hypophosphorylated, يعني let's say the normal is 5. If it has 2 phosphate groups, then it's active. If it has more than that, then it's inactive. Okay? So I want you to focus at the active and inactive states. لأنه بعدين when we're going to talk about mutations, we're going to talk how phosphorylation affects it. Okay? So active is when it's hypophosphorylated, inactive when it's hyperphosphorylated state. The importance of RB, it lies in its enforcement of G1, or the gap between mitosis and DNA replication. Okay, so the importance of the RB, it enforces the G1 so it can help it to go from G1 to S state, or the gap between the mitosis and the DNA replication. يعني it's the effect starts from the mitotic phase to the other S phase of the produced cells. Okay, مش مهم, يعني just know that it's really important in the G1 and anywhere from the M to the S phase. Okay, and يعني basically we have a cell. This cell divided, and this division occurs متى? in the M phase. انسى ال G1 والS وال G2. تمام؟ هلا من ال M دخلنا على شيء ثاني, دخلنا على على G1 جديد. G1 بيودي ل S, بيودي ل G2, بيودي لكمان M phase حتى يطلع معك كمان 2 cells. Okay, so من ال M لل S it's really important. Okay, in these gaps. In embryos, cell divisions proceed with DNA replication beginning immediately after mitosis ends. Okay. However, as development proceeds, 2 gaps are incorporated into the cell cycle. What are they telling me here? In an embryo, not much cells have been differentiated yet. So I don't really care about G1, I don't really care about G2. Here I just care about increasing the number of cells. So the cell division is going to proceed with DNA replication immediately beginning after mitosis. So هاي السل أعطتك 2 other cells, وهون مباشرة the chromosomes are going to double. Okay, مباشرة replication occurs, then again mitosis. ما راح يكون في عندنا, you won't have G1 G2, you'll just have mitosis and then the DNA replication. However, as the development proceeds, شو يعني as development proceeds? As the cells start proliferating, two gaps are going to be incorporated into the cycle. Let's imagine it this way. As an embryo, you only had the M phase and the S phase, the mitotic phase and the DNA replication phase. And the longer the cell keeps on growing and suddenly it's going to start proliferating, we're going to start incorporating phases. مين هم الphases هذول? G1 and G2 phases, okay? We're going to start incorporating these phases. Why would we do that? Because now the cell isn't just made of one type, it's made of different types. I need to regulate them, okay? Maybe another regulation, having the G1 and the G2 will help in this method. Okay, so we just incorporated the G1 and G2 into the cell cycle. So first of all, gap 1 here G1 between mitosis and the replication, gap 2 between replication and mitosis. Really easy. عشان هيك I drew it like this. Okay, so if they want to ask you, then طبعا you will start with M. Okay, so the gap between mitosis and between the DNA replication is G1. The gap between DNA replication and mitosis is G2. So please be careful in that in the exam because they're kind of like just flipped, okay? Although each phase of the cell cycle circuitry is monitored carefully and the transition from G1 to S is believed to be an extremely important checkpoint to cell cycle clock. What am I saying here? Every part of the cell cycle is important. تمام? Every step. From A to Z. لكن there are specific steps that are even more important. And the most important one is the transition from G1 to S. Once the cells cross the G1 checkpoint, لما يطلعوا من G1, they can pause the cell cycle for a time, but they're obligated to complete mitosis. شو اللي عم بيحكي لي إياه هون? Any cell that exits the G1 or crosses the G1, يعني أي cell, let's do this, G1, S, G2, M. Any cell that exits the G1 into مثلا S or G2 or even M, the cell cycle here can be paused, but it's obligated to complete mitosis. يعني خاوة عنها بعد البوز اللي بصير, I have to finish mitosis. تمام؟ لكن a cell in G1, okay, a cell in the G1 إذا ما كملت, so if it does not proceed to the S phase, we can make it enter the G0 state. In the G0 في لها نوعين, لها temporary تضل فترة معينة, so we can مثلا fix something wrong or until needed. في permanently مثل neurons and whatever, okay, في permanently. تمام? So any cell that exits G1, عدت G1, now we're talking about S, G2, M, it can pause the cell cycle whenever it needs for a specific time, لكن it will be obligated to complete mitosis. يعني eventually the cells will divide. On the other hand, any cell in the G1, here we can actually manipulate it, okay? We can make them exit the cell cycle, okay? I don't want them to complete the cell cycle. I don't want mitosis to occur, okay? So how can you control the cell cycle depending on you controlling G1? Am I going to let the cell enter temporarily, and we call this quiescence, into the G0, or permanently, and we call this senescence? Permanently, and the cells will not divide ever again in their lives. Temporarily means when needed, I'm going to call them, okay? Temporarily مثل skin, when you get a skin lesion or whatever, or when you start bleeding or whatever, here the skin cells are going to exit quiescence, okay? So they can divide and so on until the wound is closed, and after that they enter quiescence again. On the other hand, مثلا muscle cells or neurons, they are in senescence. They can never undergo mitosis again. So if any injury occurs to them, يعني good job, يعطيك العافية, خلاص ودعت. In G1, therefore, diverse signals are integrated to determine whether the cell should enter the cell cycle, exit the cell cycle and differentiate, or die. That's why G1 to S is the most important decision of a cell's life. حكينا كل الphases مهمين, all phases are important, لكن the movement of a cell from G1 to S is the most important, because here the cell has to decide, am I going to pause the cell cycle? Am I going to exit the cell cycle? Am I going to differentiate? Am I going to die? The choices that the cell needs to make when it wants to move from the G1. Okay, so G1 is really important. طيب. The RB gene is a key factor in this decision process. تمام? We must understand, and طبعا we explained in the last lecture about cyclins and cyclin-dependent kinases, and we know that cyclins are important for the cell cycle. Okay? يعني without them the cell cycle would not occur basically. تمام? The initiation of DNA replication requires the activity of cyclin E CDK2 complexes, and the expression of cyclin E is independent on the E2F family of transcription factors. Okay? Is dependent on the E2F family of transcription factors. So we have specific transcription factors called the E2F. They will cause expression of cyclin E. يعني when the E2F family هذول when they gather around the DNA, this is nothing the DNA, okay? When the E2F proteins or transcription factors gather around specific parts of the DNA, they will cause the expression of cyclin E, okay? Cyclin E, which means they will produce it. If cyclin E is produced, the cell cycle can start, okay? برضو cyclin D is important for the beginning. Early in G1, the RB is in its hypophosphorylated ايش يعني hypophosphorylated يعني active form, okay? And it binds to and inhibits the E2F family of transcription factors, preventing the transcription of cyclin E. You just told me that cyclin E is important for the cell cycle to occur, or for the initiation of DNA replication, يعني for the S phase. Okay, then cyclin E وين ضروري for the S phase. طيب ايش بدك? Again, let's draw the structure. When the E2F family, هيوم E2F family, when they gather around, they cause the production of cyclin E. Cyclin E will cause the entry into the S phase. تمام؟ قولي تمام. But before the E, there's the G1 stage. In this stage, RB protein can be active. RB protein is active. شو يعني is active? It's hypophosphorylated. تمام? It's hypophosphorylated. The active form of the RB protein شو بيعمل? It will inhibit the E2F. It will inhibit it. تقول لا, stop. If the E2F stops, then cyclin E's won't be produced. If cyclin E isn't produced, then the S phase won't be entered. And here comes the regulation of the RB protein. هاي هي الregulation تاعتها. فهمنا؟ إذا the RB protein is present in the يعني RB protein يعني what it does basically is it inhibits the E2F family. Basically it inhibits the production of the cyclin E, basically inhibits the entry into the S phase. Okay, and this occurs until everything in the G1 is completed, until the organelles increase in size, increase in number, until the cell increases in size. يعني the G1 state or the G1 phase has to make the cell completely ready for the S phase. Okay, so the G1 phase has to make the cell completely ready for the S phase. And who makes sure that the S phase doesn't happen prematurely? هو الRB gene. Okay, so the hypophosphorylated RB, it will block the E2F in two ways. It sequesters E2F. شو يعني it sequesters them? Sequester meaning that you're gonna catch it and separate it from other parts. Okay, you're gonna hold them in a specific compartment. They're present يعني هم موجودين, but they cannot do anything. High sequestering. So I will sequester the E2F and I will prevent it from interacting with the transcriptional activators. يعني it won't have the time to reach to the DNA. هضل بعيد عنه. As well as the RB recruits chromatin remodeling proteins such as histone deacetylates and histone methyltransferases, which bind to the promoters of E2F responsive genes such as cyclin E. What am I doing here? What the RB is going to do is, let's say, sort of خلينا نحكي modification to the DNA. Okay, it's going to modify the DNA, especially the histone proteins. Okay, or whatever. We're going to just modify the DNA by acetylates, by methylations to the histone. Okay, we're going to remodel the chromatin proteins. The idea here is that we're going to cause the euchromatin to become a heterochromatin, and it will not be able to be expressed. Okay, يعني if the cyclin E gene فالت لحاله, we're gonna compress it in a place that is hidden away from the E2F. Okay, instead of it being really open, it's gonna be enclosed and خلينا نحكي unreachable by the E2F transcription factors. Okay, really easy. الموضوع كتير سهل، ما بده إشي، just some focusing يعني on specific خلاص يعني. These enzymes modify chromatin at the promoters to make DNA insensitive to transcription factors. This situation is changed upon mitogenic signaling. شو يعني mitogenic signaling؟ يعني خلينا نحكي زي growth factor signaling. Okay? What happens is that when growth factors arrive, they're basically telling the cell to keep on proceeding the cell cycle. Okay? They're telling the cell to proceed on with the cell cycle. Okay, so the growth factor signaling leads to cyclin D expression, so it will activate the CDK4/6 cyclin D complexes. These complexes should be they will phosphorylate the RB. Okay, so in the G1 state, can ال RB hypophosphorylated, okay, can hypophosphorylated until growth factors arrive. Okay. When the growth factors arrive, شو بصير؟ راح يصير في عندك cyclin D CDK4/6 activation. Okay, هسه عندك activation of these. Cyclin D CDK4/6. What are they gonna do? They're gonna phosphorylate the RB. It's gonna become hyperphosphorylated. Okay, hyperphosphorylated يعني they are inactive. This means that cyclin E will be expressed. Cyclin E produces, okay, our production of cyclin E. And this means that the cell will eventually enter the S phase. تمام؟ شوف الريجوليشن قديش كبير يعني. Okay, so مهم كتير تعرف انه it's really important to know that mitogenic signaling will cancel the activity of the RB protein. So as we studied last lecture, a growth factor mutation will basically just cause multiple growth factors to stimulate the receptors, or mutations in the receptor, or whatever. So basically, the mutations in these receptors or in the growth factor themselves will cause hyperactivation, يعني more phosphorylation, which means the RB gene will not be able to govern the cell anymore. Okay? بس هاي السلافة كلها. تمام? So يعني it's really easy to understand. Now the cells are able to enter the S phase, and in the S phase they're now committed to divide without additional growth factor stimulation. يعني here the RB gene بيقول لك خلاص يعطيك العافية. Okay? I will not be able to stop you again. He had the ability to stop in the G1, but when it enters the S, it's obligate mitosis. لازم يصير obligate mitosis. Okay? تمام. During the M phase, the phosphate groups are removed from the RB by cellular phosphatases, regenerating the hypophosphorylated form of the RB because I'm entering G1 again. بكل بساطة, RB gene and I don't want it to be activated from S to M. Okay, it's going to be inactivated, يعني hyperphosphorylated. When I'm done with mitosis, I'm going to enter G1 again. I need it to be hypophosphorylated to be active. Okay, so في phosphatases, the cellular phosphatases that will remove these phosphates. E2F is not the sole target of RB, يعني RB has other targets, E2F has other activators, whatever يعني. The versatile RB protein has been shown to bind to a variety of other transcription factors that regulate the cell differentiation. RB stimulates a myocytes, adipocytes, melanocytes, and macrophage-specific transcription factors. يعني مش بس ال E2F for cyclin E, لا, it also activates other transcription factors. Okay? So the RB pathway is really important in the control of cell cycle progression when at G1. induces cell differentiation and induces senescence. كتير ضروري نعرف هاي المعلومات عن RB. Okay, so the RB pathway controls cell progression at G1, induces differentiation and induces senescence. يعني برضو لها علاقة ب G1 خلينا نحكي. Okay. So يعني this is just an image يعني كيف تحكي لك إنه growth inhibitors they will basically inactivate the cyclin D, but growth factors will activate cyclin D. This is hypophosphorylated E2F, okay? It becomes hyperphosphorylated E2F when it's inactive, okay? So here the E2F is gonna be يعني خلينا نحكي أو transcription will be activated. أما هون لما تكون hypophosphorylated transcription is blocked. تمام؟ الموضوع كتير سهل ما بده إشي. Okay, طلع كيف بيعمل histone deacetylase and histone methyltransferase. Okay, so basically just كيف أحكي إياها. So basically just block the activity of the transcription factors and so on. طيب, mutations in other genes that control RB phosphorylation can mimic the effect of the RB loss. حلو? Even anything that affects the RB phosphorylation also mimics the effect of the RB loss. What is the point of having a normal RB gene? هاي ال AA. يا حبيبي شو اللي صار؟ اوكي جبنا العيد. طيب. What is the point of having a normal RB gene? Okay, but something is phosphorylating it without the need to. Something is just making it hyperphosphorylated. عم بحط عليها كتير phosphates. There are other things that have been mutated here. Even the RB gene can be affected by other, or the RB protein or the gene can be affected by other mutations. But these mutations have to control the phosphorylation state of the RB. Okay? فكتير واضح يعني. Genes are mutated in many cancers that seem to have a normal RB genes. That's why we said not in every cancer we will find an RB gene mutation. Okay? مثلا people with breast cancer they can have a normal RB gene. طب وين المشكلة؟ المشكلة in an RB protein phosphorylation. هذا البروتين صار له mutation. It cannot actually phosphorylate the RB protein efficiently. Okay? Or it did something wrong. Or it's not phosphorylating anymore. Or it's phosphorylating too much. Okay? An example, mutational activation of CDK4 or overexpression of cyclin D, it would favor cell proliferation by facilitating phosphorylation and inactivation. يعني we're talking about overstimulation by growth factors. Okay? يعني اجى growth factor أيوه هون ارتبط. When the growth factor binds to its receptor اللي هي mitogenic pathway شو بصير طبعا excessively هون we're talking about excessively. Okay? excessively هاي growth factor excessively binding يعني أكتر من growth factor أكتر من receptor and so on. Okay? إيش اللي بصير؟ ال RB gene حكينا أنا الآن it will be phosphorylated ال RB protein it will be إيش؟ it will be phosphorylated hyper phosphorylated كمان. Okay? hyper phosphorylated hyper phosphorylated. This hyperphosphorylation هاي حرف ال B هاي كنا phosphate group. Anyways, hyperphosphorylated. This is not supposed to happen. Okay, this is not supposed to happen. Anyways, but a specific mutation caused that. Cyclin D is overexpressed in many tumors because of gene amplification or translocations. يعني lots of mutations activate the or overexpress cyclin D. Mutational inactivations of CDKIs, CDK inhibitors, also would drive the cell cycle by unregulated activation of cyclins and CDKs. يعني if you have overexpressed cyclins, يعني you have overactivated cell cycle. طب if you don't have any cyclins, يعني if you don't have مش هيك قصدي. If you have mutations that cause inactivation of the inhibitors, برضو it will cause the overexpression or unregulated activation of the cell cycle. Okay? CDKIs اللي هم cyclin dependent kinases inhibitors, they basically control the cell cycle برضو. If they were mutated causing their inactivation, يعني they won't be able to inhibit. ما راح يقدروا يعملوا توقيف لأي شيء. So everything is going to keep on going unregulated. Okay? So cyclin D is either overexpressed itself or its inhibitor is inactivated. تمام؟ Simian virus 40 and polyomavirus large T antigens, adenovirus EIA protein, and human papillomavirus اللي هو HPV E7 protein, all bind to the hypophosphorylated form of the RB. Okay? مين هم الviruses that we just mentioned. These viruses, they have the ability to bind to the hypophosphorylated form of the RB. And basically, the RB gene now won't be able to bind. لمين للE2F ما راح تقدر توقف شغله. Okay, let's also study it from here. This is the hypophosphorylated RB gene. This is active. Okay. Now, if viruses bind to this hypophosphorylated RB, مثلا HPV virus, the simian virus 40, Sim40, whatever type of virus binds, it will prevent the binding لمين للE2F site. If it prevents it, the E2F, the transcription will be activated. And ما في شيء, there's nothing stopping the E2F from doing its job. Okay, the E2F from starting, from transcripting. Okay, so it's a really easy concept يعني. Okay, حاط لكم هون يلا هانت بس صراحة أنا حتى مش مصدقها. Okay, so the RB protein will be unable to bind to the transcription factors, so functionally it's deleted, and the cells will lose the ability to be inhibited by anti-growth signals. حلوين. P53 gene اللي هو tumor suppressor gene من أهمهم ال P53, the most one that gets برضه mutated in cancers من ال tumor suppressor genes هم ال P53. نسميه the guardian of the genome. P53 induces neoplastic transformation by 3 interlocking mechanisms. Okay, هات نشوف. Activation of temporary cell cycle arrest termed quiescence, هذا الاسم تاعها. Induction of permanent cell cycle arrest بنسميها senescence, triggering apoptosis اللي programmed cell death. إذا P53 it causes neoplastic transformation by 3 interlocking mechanisms. يعني it causes خلينا نحكي the degradation of the neoplastic tissue or transformation. كيف؟ من خلال or throughout activating quiescence داخل الcell in quiescence, so it doesn't divide. برضو it can put it in senescence يعني so it doesn't divide forever. Also it will trigger cell death لحتى الcell تموت, because a cell that has an extreme or excessive mitotic ability, we don't want the risk of it being in quiescence then exiting because of another mutation, so killing it is better sometimes. So P53 is a monitor of stress. Okay, so if we see a stressed cell, I can make it, or I can guide it toward an appropriate response. What are the types of stress that the P53 recognize? Anoxia. If you remember, anoxia is complete loss of oxygen. مثلا hypoxia, low oxygen. Anoxia, no oxygen at all. Inappropriate oncogene expression. Okay, so there has been a proto-oncogene, a mutation happened to the MYC. طبعا MYC gene هو it regulates cell cycle. RAS يعني kind of the same thing. Okay, damage to the integrity of DNA. Even these types of stress and damage are recognized by P53, and then when P53 notices these damages, anoxia, inappropriate oncogene expression, or damage to the DNA, P53 is going to do its job. It's going to either cause quiescence, senescence, apoptosis, maybe quiescence then senescence, maybe quiescence then apoptosis. يعني, okay? Or in non-stressed healthy cells, يعني when the cell is healthy, P53 has a short half-life, 20 minutes. ليش? Because we don't need it. Oh, so what you're telling me here is that the P53 can extend its own half-life, okay, depending on the tissue it lies in. So if it's in a stressed tissue, the half-life of P53 would be high. Lakin if it's in a healthy tissue or a healthy cell, it will have a short half-life, 20 minutes, because of its association with MDM2, a protein that targets it for destruction. Okay, so when it's in a healthy cell, P53 is destroyed by MDM2. It basically applies a target to the P53, then destruction happens to the P53 by whatever, requinylation, whatever. يعني. When the cell is stressed by an assault on its DNA, شو بصير? P53 undergoes post-transcriptional modifications that release it from the MDM2 and decrease its half-life. إذا in a normal cell or a healthy cell هي ال P53 it will be bound to MDM2. تمام. MDM2 takes it for destruction. Okay, destruction. طيب, whenever cell damage happens on, we're talking about healthy states. Okay, healthy cells. بينما هون damaged cells شو اللي بصير؟ in damaged cells ال P53 dissociates from the MDM2. شو يعني؟ half-life increases. Okay, ال half-life basically increases. Okay, and if the half-life increases, the P53 will be able to do its job more. تمام. هاي السلافة. During the process of being unshackled from MDM2, P53 also becomes activated as a transcription factor. يعني not just it does these things, it also activates itself as a transcription factor and basically activates protein that will خلينا نحكي also suppress the growth of the cells or whatever. يعني. Dozens of genes whose transcription is triggered by P53 have been found. بس يعرف إنه lots of genes are controlled by the P53 as well. They can be grouped into two broad categories. حكينا احنا الآن when P53 causes the P53 effect is either quiescence, senescence or apoptosis. Quiescence or senescence هم عبارة عن cell cycle arrest وعادة الarrest هتكون quiescence. So we can group the genes that P53 control اللي هم dozens of genes هدول into either genes that cause the arrest or that cause apoptosis. So depending on the damage I would decide the effect. إذا كان الدمج كتير I would go into apoptosis. إذا كان الدمج قليل I'll just وزي ما حكينا إذا كان الدمج قليل basically I will tend to make the cell enter quiescence or senescence. Okay, because here the cell can actually repair itself. Okay, in the quiescence method and the cell can repair itself. When it repairs itself, when everything is good, it can move on back and basically the cell will go back to its normal state. Okay, so if the DNA damage can be repaired during the cell cycle arrest, the cells revert to a normal state. زي ما حكينا arrest, talking more about quiescence here. لكن if the repair fails, P53 will induce apoptosis or senescence. Okay, why am I talking about when I say cell cycle arrest يعني why am I focusing on quiescence? Because quiescence is revertible. You can revert quiescence, you can go back to the normal state. تمام. أما if the repair fails, if you cannot repair the damage, you don't want the cell to ever divide again. So you either kill it or you put it in permanent jail. هاي هي senescence. Okay, so let's focus on this point. لما أحكي cell cycle arrest, I'm talking about quiescence one. Okay. The manner in which P53 senses DNA damage and determines the adequacy of DNA repair are not completely understood. Okay, so we do not really know how P53 senses the damage and knows what's going on, why the DNA is bad, and why مثلا أو خلينا نحكي is the DNA repair going good or not, and so on. The key initiators of the DNA damage pathway are two related protein kinases. هم ال ATM وال ATR. We also study them in cytology. Okay, so the initiators of the DNA damage pathway, they're two related protein kinases. Ataxia telangiectasia mutated ATM, the ataxia telangiectasia mutated related اللي هو ال ATR. Patients with this disease, which is characterized by an inability to repair certain kinds of DNA damage, they will suffer from an increased incidence of cancer. Okay, so people that have people with these diseases, okay, they will have more of a chance to develop cancer. It's really simple. ليش? Because they have an inability to repair DNA. So most likely their cells are either going to die or are going to become cancerous. The types of damage sensed by ATM and ATR are different, but the downstream pathways they activate are similar. Even ATM senses a specific sort of damage. ATR senses another type of damage. لكن the pathway is the same. Once triggered, ATM and ATR, they phosphorylate a variety of targets, including P53 and DNA repair proteins. Phosphorylation leads to pause in the cell cycle and stimulation of the repair pathways. Okay? So كتير بسيط الموضوع. تمام. إذا ATM and ATR هدول اثنيناتهم they basically notice the damage and they send signals to the P53 and they send signals to DNA repair proteins. If a disease happens, okay, if basically these ATM and ATRs were mutated or something bad happened to them, the cell won't detect DNA damage. If the cell doesn't detect DNA damage, it will basically be more susceptible to developing cancer. Really simple. Okay? طيب. P53 mediated cell cycle arrest may be considered the primordial response to DNA damage. Okay? It occurs late in the G1 phase and is caused mainly by P53 dependent transcription of the CDK1 and CDKN1A اللي هو P21. A P21 هو CDK inhibitor. Okay? Okay, it's a cyclin dependent kinase inhibitor. Really important to know that. Okay? So P53 high وين بيجي الإفكت تاعها? The effect is late in the G1 phase and it's mainly by P53 dependent transcription of the CDKI اللي هي cyclin dependent kinase inhibitor اللي هو P21. The CDKN1A gene inhibits cyclin CDK complexes, okay, and prevents phosphorylation of the RB essential for cells to enter G1 phase. Okay, so basically it prevents the inactivation of RB. مين هذا اللي بيعمل هيك؟ P53. So P53 notices the damage, as a result activates some CDKIs, CDK inhibitors. These inhibitors will inhibit cyclin CDK complexes, and these complexes will and this inhibition of these complexes will prevent phosphorylation of RB. يعني they will keep the RB gene active. شو يعني active؟ يعني basically it will the cell will not خلينا نحكي proceed G1. Okay. تمام. Such a pause in cell cycling gives the cells time to repair the DNA damage. P53 also helps the process by inducing certain proteins, like the growth arrest in DNA damage 4-5. Okay? It basically just helps in DNA repair. If DNA damage is repaired, then the P53 upregulates transcription of MDM2. شو بيعمل؟ MDM2 will bind to P53, it will decrease the half-life P53 and destroy P53, and it will relieve the cell cycle block. If the damage cannot be repaired, حكينا إما senescence or apoptosis. More than 70% of human cancers have a defect in this gene. Lots of cancers have defects in P53 gene. اللي هو حكينا one of the most common genes برضو. And the remaining malignant neoplasms have defects in genes upstream or downstream of P53. إذن هاي P53 gene المutations إما بيكونوا عليه 70% and the others are either upstream or downstream of this gene. Okay? So يعني موضوع واضح ما بدوش كتير حكي. Anyways, homozygous loss of the P53 gene is found in virtually every type of cancer. Homozygous loss يعني you're losing both normal types of the P53. Carcinomas of the lung, of the colon, of the breast, okay? Yeah. Less commonly, some individuals inherit a mutant P53 allele. This disease is called Lifraumeni syndrome. We studied it like I think a lecture or two ago. يعني it's not really that يعني difficult of a disease to know. Okay, ارجعوا له يعني شوفوا بالجدول شو مكتوب. So a mutant P53 allele is inherited Lifraumeni syndrome. Inheritance of one mutant allele predisposes individuals to develop malignant tumors because only one additional hit is needed. يعني نفس موضوع ال RB and the two genes must be mutated فلازم تتحول ل AA. Okay, نفس الموضوع اللي شرحناه قبل شوي فكتير واضح. Patients with Lifraumeni syndrome have a 25-time greater chance of developing a malignant tumor by age 50 compared with the general population. In contrast to patients who inherit a mutant RB allele, the spectrum of tumors that develop in patients with Lifraumeni syndrome is varied. The most common types are sarcomas, breast, leukemia, brain tumors, and carcinomas of the adrenal cortex. Okay? And this is the picture يعني clearer خلينا نحكي لأن إذا ما كانت الصورة فوق واضحة. And we explained it and hopefully everything was A-okay. Good luck, doctors, and now let's solve some questions. Okay. أول واحد, what is retinoblastoma gene known for? It's basically a cancer suppressor gene. What is the impact of hyperphosphorylated? حكينا it will inactivate RB. If it's inactivated, you can progress from G1 to S. Okay? So basically, it promotes cell cycle progression. Even D برضو. Sorry. What happens to P53's half-life during cellular stress? The half-life will increase. So A. تمام? Which types of cancer have been associated with the loss of a normal RB genes? حكيناهم اللي هم breast cancer, lung cancer, and bladder cancer. What are the 2 hits required for the development of retinoblastoma? حكينا 2 hits and 2 mutations على مين؟ على ال RB genes. So 2 mutations in the RB gene. What role do cyclin E CDK2 complexes? They basically خلينا نحكي they stimulate DNA replication initiation. Okay, they cause the initiation of the DNA replication. Okay. What types of tumors are commonly associated with La-Framini syndrome? Sarcomas, breast cancer, leukemia, brain tumors, and adrenal carcinomas. What percentage of retinoblastomas are sporadic? اللي هي 60 to 40, 60 يعني sporadic و 40 familial. What triggers the transition from G1 to the S phase in the cell cycle? أكيد اللي هي presence of growth factors. And this was it for today's lecture. I hope you enjoyed it, and don't forget to show your support by like, subscribe, maybe comment something good. And good luck, doctors. بالتوفيق.